Hi everyone, and thank you for joining me for my thesis defense, which was entitled Relationship of Blood Parameters and Fertility-Related Traits with Age and Feed Efficiency in Young Beef Bulls. I would first like to thank my external examiner, Dr. Kim Aminsky, for being present, as well as the Graduate Studies Coordinator, Dr. Deanne Pedersen, and my advisory committee, Dr. Yuri Montagnoli, Dr. Alan Ferdin, and Dr. Mariana Diamone. During this presentation, I'm going to be going through a typical scientific outline, during which I would describe both of my studies and finish up with my master's experience. So feed efficiency is an important trait for the beef industry as it results in a decrease in its environmental impact and an increase in its profit. However, age is an important factor to consider, especially when evaluating young bulls for feed efficiency, as age has been shown to be associated with feed efficiency, body composition, blood parameters, and sexual development. Also, fertility-related measures offer the potential to act as both indirect indicators of feed efficiency and fertility. However, because these measures changes during sexual development, care must be taken. Such measures include sexual hormones, such as testosterone, which increase with puberty in young bulls. The measures also include scrotal circumference and testes pixel intensity, and both of these increases with sexual development, as shown here by the constant increase in scrotal circumference with sexual maturity. Moreover, semen quality increases with age, and here you can see the drastic increase in progressive motility following puberty of young bulls. Also, such measures have been shown to be associated with feed efficiency, including scrotal temperature, scrotal circumference, testes echogenicity, semen quality, seminal proteinomics, and testes microscopy. So because all of these are interrelated, there is a potential to work on each of these in order to improve the overall profitability of the beef industry. So for example, if we're able to increase feed efficiency and reproduction of young bulls, we would most likely be able to produce progeny that are also feed efficient, which could lead to an increase in the competitiveness of the beef industry with other livestock species. Then, having bulls that reach puberty at a younger age but still have a good reproduction, these bulls would most likely sire daughters that also reach puberty younger and have good reproduction, which could lead to an increase in the reproductive success of the industry. Now, being able to understand and control the effect of age on feed efficiency measures would allow us to use indirect indicators of feed efficiency, such as blood parameters, which would lead to a drastic decrease in the time needed to assess feed efficiency and also in the labor. Altogether, these would lead to a decrease in the environmental impact of the beef industry and also an increase in its profit. So, cattle that differ in feed efficiency are known to also differ in metabolism, and metabolism is closely related to reproduction, which may explain the recent discovery that reproduction also vary with feed efficiency. Also, age is an important factor that affects feed efficiency, metabolism, and reproduction. Therefore, the hypothesis of the study is that animals that differ in feed efficiency and age will also differ in their metabolic and reproductive measures, as well as in their sexual development. The overall objective of this thesis was to assess variation in blood parameters and fertility-related traits with age and feed efficiency over time and on a single-day measure. The first study of this thesis is entitled Association of Blood Parameters with Age, Feed Efficiency, and Sampling Routine in Young Beef Bulls, and this paper is currently under review at the Journal of Livestock Science. The objective of this study was to assess the variation in blood parameters such as blood hormones and blood metabolites with age and feed efficiency using two residual phenotypic models, with one adjusting for age over time and on a single day. For this study, 32 yearling crossbred bulls were assessed. Bulls were housed at the LR Beef Research Center and were mainly Simmental and Angus of breed. Bulls were fed during a 112-day feeding period during which they were fed a high moisture corn diet, and daily feed intake were measured using automated feeding system. Body weight and body composition traits were assessed every 28 days. Body composition traits included back fat and rump fat thickness, rubiuria, and marbling score. Residual feed intake was used as our measure of feed efficiency, and the first model adjusted for body size and body composition traits. And then for the second model, we also included adjustment for age. Blood was collected via jugular vein puncture during a 32-day blood sampling period. And blood was also obtained during bleeding, during slaughter, during which bulls were slaughtered in group of 4 at 8 different days. The blood parameters that were measured included metabolic products such as carbon dioxide and acetate, ions such as potassium, proteins such as alkaline phosphatase and brain-derived neurotrophic factor, sexual hormones such as testosterone and luteinizing hormone, metabolic hormones such as T3 and leptin. For the statistical analysis, we first assessed normality, and then bulls were divided into age group and feed efficiency group using the 2 residual feed intake models, and there was 16 bulls in each subcategories. Means comparison over time was obtained using proc mix, while mean comparison at slaughter was obtained using proc GLM. In both cases, we adjusted for the breed effect. Now for some results. We observed lower testosterone in younger bulls, which agree with the literature as testosterone increases with puberty in young bulls. We observed higher CO2 in efficient bulls, and this further supports results showing differences in stress response between feed efficiency groups, as CO2 levels vary with respiratory rate following a stressful event. The higher acetate observed in the efficient bulls may be related to the association between acetate and feed intake. Has higher acetate in the rumen and blood of cattle is associated with depressed feed intake. We observe higher testosterone in efficient bulls, and other studies have described a positive association between feed efficiency and testosterone. However, in this case, the difference may also be due to the variation in age between feed efficiency groups, as once we included age in the model, there was no longer a difference. Looking at T3, feed efficient bulls had lower T3, and this may be due to the association between T3 and energy expenditure, and may explain the higher feed intake in inefficient bulls. On the day of slaughter, we still observe lower T3 in efficient bulls. This suggests that T3 may be robust to variation in sampling routine. And then looking at brain-derived neurotrophic factor, we observe higher BDNF in efficient bulls. And BDNF has been associated with feed intake and energy homeostasis, suggesting that in the future, BDNF may become an important indicator of feed efficiency. So in conclusion, blood parameters did vary with age and feed efficiency, and once age was added to the R5 model, it impacted the association of certain blood parameters with feed efficiency. The difference between feed efficiency groups for BDNF was robust to variation in age, while the difference between feed efficiency group for T3 were robust to variation in age and sampling routine. So because we observed interesting relationship in our first study, with some of them also being important for sexual development, we decided to further investigate the relationship of age and feed efficiency with sexual development and fertility-related measures in young beef bulls. And this paper is currently under preparation to be submitted to the Journal of Animal Reproduction Science. The objective of this study was to look at the variation in blood parameters and fertility-related traits with age and feed efficiency over time and on a single day. For this study, we had access to 2 populations of bulls, which were classified as either British or continental, and the bulls were yearlings. Bulls were assessed over an experimental design at the start, middle, and end, with the end being around the time of breeding soundness evaluation. For all assessments, we looked at body composition, body weight, scrotal circumference, testes ultrasonography, and blood hormones. And then at the end, we also assessed blood for the complete blood cell count analysis, scrotum thermography, and we collected semen for semen quality. Bulls were fed for 112 days, and daily feed intake was measured using a automated feeding system. Maritime's bulls were mainly fed grass silage diet, while Aurora bulls were mainly fed a high moisture corn diet. Similar to the first study, body weight and body composition traits were used in the calculation of residual feed intake, in which we adjusted for body size and body composition, as well as the different bull population. We measured scrotal circumference and testes ultrasonogram, as both of these traits are associated with testicular growth and testicular development. And then the image obtained from the ultrasound was analyzed by computing proportionate squares using the scrotal circumference of each bull, and the pixel intensity was measured within each of these squares. Infrared images were obtained using an infrared camera, and then the images were analyzed for scrotal surface temperature over the whole scrotum and scrotal surface temperature at the base and apex of the scrotum. We collected semen using through electroejaculation, and semen was immediately analyzed for sperm motility under the microscope. Then we also took a part of the ejaculate and stained it to assess sperm morphology, such as head defect, midpit defects, loose heads, and tail defects, and also sperm viability. And the reason why we look at all of these parameters is in order to assess the breeding potential of the bull, and also to infer about the stage of sexual development. We collected blood through jugular vein puncture and look at hormones such as testosterone, and we also did a complete blood cell count analysis to assess the health status of the bulls. For the statistical analysis, normality was assessed similarly to the first study. Then we divided our bulls into age and feed efficiency groups according to the distribution of age and residual feed intake. Mean comparison over time was performed using PROC-mix, while mean comparison on the day of the breeding soundness evaluation was performed using PROC-GLM. In both cases, we adjusted for breed and population effect. If the trait that was under analysis is influenced by environmental temperature, we also adjusted for temperature humidity index. Such traits included scrotal circumference, testes ultrasonogram, and scrotal surface temperature. And when we were comparing feed efficiency groups, we also adjusted for age. We also performed analysis of variance to compute residuals, and these residuals were used in the calculation of person correlations. We also used these residuals for partial least square analysis, where we created 6 different factors that were then used in the partial regression analysis. Now for some results. When looking at age group, as expected, we observed lower feed intake, lower body weight, lower back fat thickness, and lower rhibiuria in younger bulls. We also observe a smaller scrotal circumference in younger bulls over time, which agrees with the literature, and then a bit different than what was expected. We observe a trend towards significance towards a higher pixel intensity in the younger bulls. There was no difference in blood hormones between age group, and differently than the first study, although the curves appears to be different, there was no difference in testosterone levels between age group. Scrotal circumference was still smaller in younger bulls on the day of the breeding soundness evaluation. In general, we observed higher scrotal temperature in younger bulls, and this suggests that these bulls may have more metabolically actively growing testicular tissue. In general, younger bulls had lower semen quality, which was expected as semen quality improves with age. And then looking at feed efficiency groups, there was no difference in body composition traits between the two groups, which is positive, showing us that our model was properly computed. Younger bulls had lower scroll circumference efficient bulls had lower scroll circumference over time, and although there was no difference in mean pixel intensity between the groups, we did observe a trend toward lower maximal pixel intensity in efficient bulls over time. There was no difference in blood hormones, and differently to our first study, although the curves appear to be lower in efficient bulls, there was no statistical difference in T3 between efficiency groups. On the breeding soundness evaluation, we still observe smaller scroll circumference in efficient bulls, and although there was no difference in pixel intensity between the groups, this was expected, as at this stage most bulls were post-pubertal. In general, we observed lower scroll surface temperature in efficient bulls, and this suggests that these bulls may have less metabolically active testicular tissue and may allocate less energy towards testicular growth. Looking at semen quality, we observe a trend toward higher sperm head defects in efficient bulls, and this agrees with other studies and suggests differences in semen quality between feed efficiency groups. We observe positive correlation between T3 scroll circumference, scroll surface temperature, and sperm tail defects, suggesting an important role of T3 with testicular development and function. Looking at the variance analysis, when only looking at the whole population in terms of age, the most important factor explaining variation in age was scroll circumference. However, when only looking at the younger bulls and the older bulls, the most important factor was semen quality. In terms of the whole population, in terms of feed efficiency, the most important factor explaining variation in residual feed intake was scrotum temperature. And then when only looking at the efficient bulls, the most important factor was semen quality, while for the inefficient bulls, the most important factor was the CBC traits. So in conclusion, younger bulls presented characteristics of sexual immaturity, while efficient bulls displayed features of delayed sexual development. Scroll circumference explained considerable variation in age, while scroll tomography explained most of the variation in feed efficiency. So in scroll tomography and scroll circumference should be considered when testing bulls in the context of feed efficiency. So in general, we observe an important difference in testicular development between age and feed efficiency groups, and a lot of organs and processes work together for the onset of puberty and sexual development in bulls. These include the hypothalamus and the pituitary, which work together to increase the secretion of testosterone by the Leydig cells and the secretion of thyroid hormones by the thyroid gland. And in general, we observed lower testosterone in younger bulls, which was expected and may be related to the features of sexual immaturity observed in these animals. Then we observed positive correlation between T3 scroll circumference, scroll temperature, and semen quality. And in general, lower T3 was observed in the efficient bulls, which may be linked to the delayed features of sexual development and testicular development observed in these bulls. So for future studies, it would be interesting to look at the similar assessment, but over various physiological stage in bulls and over the circadian pattern. And it would be interesting to also look at the specific time of puberty and sexual maturity in bulls of different feed efficiency, and also at the microstructure and molecular aspect of bull reproductive organs. And altogether, these studies will allow us to develop protocols that could help us find bulls that are both feed efficient and have good reproduction. So I would like to thank my supervisor, Dr. Yuri Montagnoli, my committee members, all the academic support I received, my coworkers, and the technical staff at both firms, as well as all the funding agencies. And over the past 2 years, I've spent many hours working with beef bulls, and I was lucky enough to also be working with the community by helping out through the testing of commercial bulls and helping out producers through extension work. And I was lucky to do an oral presentation at the Canadian Society of Animal Science, do an oral presentation at the 66th EAAP conference in Warsaw, Poland. And also, I was fortunate to receive a scholarship with CMEX through NSERC, and that was a great opportunity for me to work within the industry. And I'm very excited to be presenting my work at the conference for the Association for Applied Animal Embryology this summer in France. Thank you.